Good morning, everyone. My name is Dr. Marvin Sigal, and I am here to give a teaching demonstration for Craven Community College. And the topic of my teaching demonstration today is the mechanism, technique, and significance of Gram staining. So let me begin. It's important in biology, and especially in medicine, to be able to identify a particular species of organism. And for large organisms, that's pretty easy. One can identify, for example, trees by the shape of the leaf or other anatomical features like the bark. Birds, for example, are easy to identify because of the coloration of the feathers, the shape of the bill, also the shape of the entire bird can tell one species from another. Even small organisms like tiny little arthropods that need to be put under a microscope, one can identify the species by the markings on them, also by particular anatomical features such as hairs and setae that are on the mites under a microscope. However, bacteria are not easy to identify. They are extremely small, very, very much smaller than mites. And even at high power under a microscope, they would only appear to be like little dots and dashes if viewed under the microscope. So it's important to have a way in order to first identify that a particular specimen, tissue specimen, has bacteria in it, and then to go on and be able to identify what type of bacteria it is. So to start off, one would use a simple stain. And a single type of stain is used to basically show the size, the shape, and the arrangements of cells that would be found as bacteria. The shape would be either spherical shaped or they would be rod shaped type bacteria, and they could also be spiral shaped bacteria. Also, the bacteria can either be found singly, individually, or they can be found in clusters. So again, to first identify a particular tissue sample as having bacteria is the first step, using a simple stain. But then once we've done that, it's important to then be able to identify one type of bacteria from another. In other words, you could have two different types of spherical bacteria, but they are of a different species, and one of them may be extremely pathogenic and cause a serious disease, and other ones may not be. They may be benign and natural. So in this case, one would use what's called a differential stain. And a differential stain allows you to distinguish two different types of bacteria, and the stain that has been used in microbiology has been called the Gram stain technique. The Gram staining technique was devised by a scientist by the name of Hans Christian Gram, and he lived in the late 1800s into the early 1900s. And in the 1880s, he developed a technique by which one could stain a tissue sample or a biopsy or autopsy sample, which is what he was doing, and be able to use this technique to identify bacteria that may have infected that tissue. So what he did was he developed this technique, and he used different stains in order to differentiate one type of bacteria from another. By doing this, he was able to distinguish what he called gram-positive and gram-negative bacteria. It was also determined that there are other bacteria that are called gram-variable and other bacteria that are called gram-non-reactive. And these four types of bacteria could then be distinguished using this technique. Bacteria have cell walls, and the cell walls contain a compound called a peptidoglycan. And gram-positive bacteria will contain much of this and many layers of this peptidoglycans. And within this layer, there is another molecule that is called teichoic acid. Gram-positive bacteria will contain this teichoic acid, and gram-negative bacteria will not. So what happens is, if one uses two different molecules, two different stains or dyes, one of which is called crystal violet, and the other one is called iodine, those two molecules will bind with the teichoic acid and they'll form a very complex molecule within the cell wall. And once it's done that, especially if it's gram-positive, there is so much of it that it's very difficult to wash away those complexes once they've been stained. And that's the basis for how this gram staining technique occurs. You would first stain the bacteria using these two stains, and then you would wash it away basically. And if there's no teichoic acid, then this iodine, this crystal violet and iodine will not be bound and form a complex with the molecule. And so therefore, there would be no coloration left over when it's done. However, if the bacteria is gram-positive, the large amount of these complexes are not easily washed away, and as a result, there will be a coloration to those bacteria that would distinguish it basically from those that are gram-negative and do not have that complex with them. Now, what is the technique that is used in the laboratory in order to determine whether a specimen is made up of gram-positive and/or gram-negative bacteria? First thing one does is prepare a smear, a slide with bacteria on it, and then one would go through the following procedures. First thing to do would be to stain or to apply the crystal violet to this slide. And this, you would stain it for about a minute, and this crystal violet is purple, and this would stain all of the bacteria, be they gram-positive or gram-negative, with this dark crystal violet stain. After it's been stained, you would then drain the stain and then rinse it with water. Secondly then, one would then take another stain, iodine, and apply it to the smear. And what would happen is this iodine then binds with the complexes and produces what's called a mordant, and this mordant acts like a binder. It sort of sets the stain and makes it more difficult to remove. So you stain it with the iodine for about a minute, and then after that you would drain and rinse that away as well. Okay, once you've done that, you would then want to decolorize your specimens, and this you would use alcohol in order to do this. You would take alcohol, and just with a quick rinse, that would help remove some of the stain, and then immediately after doing that again, you would rinse that with water. Once you're done with that, you would then once you've removed that, the gram-positive bacteria will hold on to the crystal violet stain, and the gram-negative bacteria will not. So what you have at this point in your procedure is that you have only the gram-positive bacteria that have been stained a purple color. Okay, the gram-negative would have no color to it at all, but you still want to be able to identify those gram-negative bacteria. So you would then continue with the procedure as follows. Once you've done that, you would then take another dye. It's called safranine, and it's a red dye, and you would then apply this to the smear and allow it to set for like about a minute, 30 seconds to a minute. And when that happens, the safranine, the pinkish reddish safranine dye will then bind again to both of the bacteria, but because the gram-positive is very dark, you won't see that lighter pinker safranine stain. However, the gram-negative bacteria, those that before were clear and pretty much invisible under the microscope, those have been now stained a light pink stain. Okay, and when you're done with the staining, you would then drain the stain, you would rinse it, and then you would blot that in order to remove any of the excess liquids. Once you've done that, then you now have your finished product. And if you were to look at this under the microscope, what you would see then is that the gram-positive bacteria would be a dark crystal violet color, a dark purple color, and the gram-negative bacteria would then be a lighter pinkish saffronine type of color.